Okay, that's it. Okay, yes. So specifically, what I'm going to tell you about today is our work on studying injury-induced reprogramming of retinal glia into neurogenic progenitors, which can then replace cells that have been lost due to injury. Now, the retina is really an extremely tractable and accessible part of the central nervous system and contains six major types of neurons, and really has highly evolutionarily conserved anatomy, very tractable, is really in many ways the most an extremely tractable region of the central nervous system and a great model for studying the brain and the spinal cord. So the retina consists of six major types of neurons, one type of glia, and really the star of the show today is going to be the mutaglia, which are the only cell type or the only glial cell type that arises from retinal progenitors during development. Now, mutaglia closely resemble retinal progenitors in many respects. Their radial morphology, their gene expression profile, and their ability to potentially their ability to proliferate and their ability to proliferate following injury. Now, most forms of of inherited blindness result from the degeneration of specific cell types within the retina. So, for instance, loss of the rods, which sense dim light, or the cones, which sense bright light, are what causes blindness in diseases such as retinitis pigmentosa or macular degeneration. Likewise, loss of the retinal ganglion cells, which send information out through the optic nerve to the brain proper, leads to glaucoma. So there's tremendous interest in potentially using these cells as a means of replacing these cells in models of retinal injury. Now, before I move on, I want to mention the retinal interneurons. Photoreceptors project to bipolar cells, amacrine cells, in turn modulate the bipolar to ganglion cell synapse. I'll be saying quite a lot about both those cell types later. Now, one of the reasons that there's so much excitement in using glia as a means of reprogramming glia as a means of regenerating neurons lost to disease is the fact that most vertebrate species do this already. So, for instance, most cold-blooded species, and the zebrafish really is an exemplar here, the glia essentially act as quiescent injury-induced neural progenitors. So that, for instance, in the zebrafish, if you kill the photoreceptors with bright light, you kill the interretinal neurons with NMDA excitotoxicity, the glia will reprogram within a couple hours to a neurogenic state. They'll start proliferating within about 48 hours. And then over the course of days to weeks, all of the lost cells of the retina are regenerated. And this can happen again and again and again, up to half a dozen times before there's any obvious defect. Now, this is a real superpower, but it has been lost multiple times over the course of evolution. It's been lost entirely in adult warm-blooded species, birds and mammals. Although some species, such as post-hatch chikhea, retain a very limited ability to make neurons following injury during the first couple of postnatal weeks. Now, unfortunately, all vertebrates, mice, humans, everyone we've looked at, the glia do not make any neurons following injury. Instead, what happens is that the glia reprogram to an activated or gliotic-like state, and then over the course of several days, return to a resting state, at least following an acute injury. Now, again, this is a case where we would really like to be more like a fish. So what strategies have been used to induce reprogramming of mammalian mutaglia? So most of the effort has really gone the Yamanaka-Wernicke way, simply to take neurogenic factors, usually transcription factors, and then misexpress these, either using transgenics or viral vectors, to convert the glia to a neurogenic progenitor. And there have been several groups, particularly Tom Ray's group at the University of Washington, that have had considerable success doing this with factors such as ASCL1. So another method is to turn this on its head, to simply ask what is inhibiting the ability of mammalian glia to regenerate and block those inhibitory cues. This is going to be the major focus of my talk, identifying these and disrupting. And then finally, once you've made a glia into a neurogenic progenitor, you then have to get it to make the cell types that you want. And there, typically, the strategy has been to use transcription factors that drive the development of specific cell types and misexpress them. And I'll have to say there's been much less progress on that. I'll touch briefly on it. So my lab has been using unbiased omic approaches, really before the term was even used, where to really identify genes that are dynamically expressed over the course of development, disease, and regeneration, using a comprehensive and unbiased expression profiling approach. Doing this for about 25 years. Doing this initially using just whole retina, bulk dissected cells, then later isolated progenitors. These approaches were powerful, but they were low resolution and missed a great deal. About 6-7 years ago now, with the development of droplet-based single-cell RNA sequencing, it really became possible to visualize essentially any dynamic process at the level of individual cells. And this, in many ways, was really comparable to the development of the Hubble Space Telescope relative to ground-based telescopes in terms of its level of resolution. You can see everything that's happening almost in real time. Now, together with this, one can also use a parallel approach called ATAC-seq, or Assays for Transposon Accessible Chromatin DNA-seq, to analyze gene regulatory networks. The way this works is that it simply takes a bacterial transposase, TN5, along with a couple of DNA linker sequences with a transposase attachment sites. These can then be used to integrate essentially randomly into open chromatin anywhere across the genome. And this allows you to visualize not only the position of nucleosomes, chromatin conformation, but much more interestingly, to visualize transcription factor binding. Essentially, when a transcription factor is bound to a specific site, this is protected from transposon integration. And you have a little footprint, as shown here. Now, if you know the expression pattern of transcription factors in the cell of interest from the single cell seq data, and you have an idea of where the preferred target sites for that transcription factor, by developing a fine-grained, fine temporal series, one can essentially reconstruct gene regulatory networks associated with any dynamic process. And in this case, the focus is response to injury. So what I'm going to tell you about is first, published studies where we have used this approach to identify gene regulatory networks that are activated following injury in both regeneration competent and incompetent species. I'll then tell you about, I'll then present unpublished data that shows how we have taken this further and identified additional components of the network. And then finally, I want to discuss some of the potential promises and pitfalls in applying these findings to induce regeneration and glial reprogramming in vivo. So first, the published work. So as part of a multi-lab study funded by the Audacious Goals Initiative of the National Eye Institute, we set out to profile changes in gene expression in whole retina in 3 different species with very different regenerative competence. So zebrafish are champion regenerators. Mice basically have no ability to regenerate at all, and post-hatch chick have a very limited neurogenic competence. We analyzed whole retina, or either whole retina or FACS-isolated muloglial cells. We started this out in the bulk sequencing era, and then analyzed 2 different modes of damage. So NMDA injury, where we kill the interretinal neurons with excitotoxicity, light damage, where we kill the photoreceptors. In the case of zebrafish, it's actually possible to induce glia to become neurogenic in the absence of injury with a Notch inhibitor and TNF-alpha. And then we analyze this at multiple time points, starting from essentially 0 to 48 hours post-injury, and generated both single cell and bulk RNA and ataxia. What we do then is simply to integrate these data sets and then compare changes across the 3 species. We want to identify first common changes that are really just reflect a general cellular response to injury, and then species-specific differences that are either positively or negatively correlated with regenerative competence. We can then put all this together to identify gene regulatory networks, identify key transcriptional regulators, and then target these functions. So that's the work. This is some of the single cell data from the 3 species. This is basically, these are mouse, fish, and then chick treated with NMDA. And the different cell types here are color-coded. We can readily identify all of the major cell types. The glia are highlighted here in red. So we can readily distinguish between resting and activated glia, and in the case of the fish, we can identify some progenitors as well. What happens, the glia, the behavior of the glia following injury is very different in all 3 species. What we can do in each species is digitally isolate the glia, and then we can ask what changes over time, or what changes in response to injury using a pseudo-time analysis, where we simply just take each of the cells. So just each of these, just to clarify, each of these are two-dimensional T-SNE plots, where we've essentially taken 15 to 20 principal components of variation, crushed them down to 2 using some machine learning magic I don't really understand. And then we're essentially displaying like cells together with like. So the closer these dots are together, the more similar they are. So again, these are the glia. We know that based on expression of cell-specific markers. And here, these are glia that are essentially, these are glia, and this is how the glia change over time. So from essentially most to least different, these are the resting glia. In the mouse, the glia that are most transcriptionally different from the resting glia are actually seen only 3 hours following injury. They then gradually return to a resting state over the next few days. Now, in the fish, it's a very different story. So here again, we have resting glia. These glia transition to an activated-like state, which in many ways resembles that of the mouse. But then, while a few seem to revert to a resting state, these arrows represent the direction of change or essentially the direction of movement using a technique called RNA velocity. Most then go on to reprogram to cells that transcriptionally are effectively identical to progenitors that you see in the developing retina. Now, chick are somewhere in between. 24 hours following injury, all of the glia are reactive. Most of them return to a resting state. A subset then go on to reprogram to a proliferative state. So by putting all this data together, so this RNA-seq together with the ATAC-seq, we are able to identify 3 super networks in the regeneration competent and incompetent species. Now, there are some key similarities and differences between these. So in both the mouse and the zebrafish, there are two networks, one that's active in resting glia, the second that is active in activated glia or in gliosis. So these consist of transcription factors that cross-activate one another's expression. So the network is auto-activating. Then transcription factors in this network inhibit genes in the gliotic network and vice versa. So you have two networks that are auto-activating and cross-inhibiting. This kind of bistable network you see all over the place in biology. This is a variation on a theme. Now, what happens next, though, is very different. So this gliotic, so cells in this, once in the zebrafish, when they enter gliosis, then are predicted to rapidly reprogram to a proliferative state, rapidly and essentially unidirectional. Now, in the case of the mouse, this third network has an opposite effect. It acts to restore a resting state. So this consists of genes, most of which that are expressed in progenitors, whose expression is rapidly downregulated following injury and then overshoots baseline levels by about 6 to 8 hours following injury. And then, so this essentially actively stops regeneration, reprogramming in its tracks, and returns cells gradually to a resting state. So there are there are several key predictions here. Number one, we predict that cells will have to transition through this activated state in order to reprogram. So you block transition from rest to activation, you would block regeneration. Second, there may be something fundamentally different about the genes in the mouse, the mammalian versus the fish activated network that promote versus constrain regeneration. And finally, that regeneration is actively inhibited in mammals. So we set out to test this. So first, this is a collaboration with David Hyde at Notre Dame University. We inhibited the activity of multiple transcription factors in the gliosis network seen in the zebrafish, either with morpholinos or with drugs, and that blocked reprogramming. Second, we found that transcription factors associated with the classic inflammatory response, particularly NF-kappa B and also STAT3, were much more strongly activated in mouse. They're activated to a limited extent in fish, but much higher in mouse. We disrupted these genes both in chick and also in mice that were transgenic mice that were overexpressing ASCL1, and thus were given a measure of neurogenic competence. That in turn blocked reprogramming. Now, finally, I want to focus on the genes in the restore rest network. So these include 3 members of the NFI family: NFIA, NFIB, NFIX. Now, we had previously studied these in the context of development. So in the developing retina, we see a clear molecular distinction between progenitors in the early and late stages of neurogenesis. NFI genes are highly expressed in late stage progenitors, and their expression level gradually rises as development proceeds and is essentially maximal in resting glia. When we misexpress the NFIs in neonatal progenitors, we see that this promotes the formation of late-born cells, inhibits the formation of earlier-born cells, and drives cells out of cell cycle. And conversely, loss of function in retinal progenitors has the opposite effect. No late-born cells. You see progenitors persisting for up to 2 months postnatally, and they generate only photoreceptors. So these seem to act as general indicators that this is a general timekeeping signals and tell the cell that it's late in development, it's time to make glia, it's time to stop dividing. We were then able to use a combination of ChIP-seq and RNA and ATAC-seq of NFI triple mutants to really figure out how the NFI factors work. They act as pioneer factors that bind to cis regulatory elements that are specifically associated with late-stage progenitors of neuroglia. They open chromatin, they allow other transcription factors to bind, and in turn promote gliogenesis. So these are genes that have a well-characterized function in development of glia and in preventing proliferation. So what do they do in the adult? So the way we addressed this was to use a tamoxifen-inducible Cre recombinase along with an NF5 triple mutant, and then a conditional mutant, and then a Cre-inducible SUN1 GFP fusion protein under a constitutive CAG promoter. So what happens when we activate the Cre using tamoxifen is that we simultaneously delete all three NF5 factors in glia and selectively and permanently activate the SUN1 GFP, which labels the nuclear membrane. So in this case, these are 2-month-old animals. We do the tamoxifen injection. We wait 7 days. We injure with NMDA, and then we collect 2 weeks after that. Now here's a control. These are animals that have been treated with NMDA and also FGF2 and insulin. These are the glia. And as you can see, the glia are sitting here just below the bipolar cells that express the transcription factor CRX, and just above the HUCD positive amacrine cells. What happens when you knock out the NFIs, however, is that the glia, first of all, start to move. They start to migrate radially, and they now start, some going up into the photoreceptor layer here, and they start to express both CRX and QCD. So markers of photoreceptor bipolar and amacrine cells. And this is never seen in the control. Now, what's going on at the molecular level? We're able to characterize that using single-cell RNA-seq. So here, same protocol, but here we've actually sorted out the glia. These are the control cells. They all look like neuroglia, as do about 85% of the mutant cells. However, what we're able to see is a cluster of cells that are small cluster of cells that are proliferating. These just look like proliferating neuroglia. And more interestingly, a cluster of cells that look like neurogenic progenitors that are actually not dividing, but are giving rise to bipolar-like and amacrine-like cells. And we can visualize these morphologically by infecting with basically a cytoplasmic filling and cherry expressing virus. These are the glia, but we can see a number of cells that have bipolar cell-like morphology with dendrites pointing towards the retinal pigment epithelium and amacrine-like morphology with dendrites pointing the other way. By 2 months, we're starting to see cells expressing a number of protein markers of bipolar cells. So just to sum up this part of the talk, this is all published. By using integrated multi-omic analysis, we're able to identify gene regulatory networks that control regenerative competence and that actively repress it in the mouse. And we've shown that the NFI factors actively are part of this repressive network. Now, we can really, I think, do much better. So this is an important advance, but we're really not where we need to be. So we're still basically we're not making the cell types we want. We're making bipolar and amacrine cells. We want to make photoreceptor cells and ganglion cells. We also want to promote neurogenesis and proliferation. We see very little proliferation, and at best, about 15% of the glia convert. So we've been focusing really on methods to both enhance neurogenesis and to further inhibit these inhibitory factors, and thereby promote the formation of the cell types we want. And ultimately, we want to do this in wild-type animals. We want to do it with one virus, and we want to do it with one cocktail of small molecules. That's the dream. So what I'm going to tell you next is all unpublished, and really our efforts to do each of the following. First, to promote neurogenesis by misexpressing transcription factors that promote neurogenic competence. So we've done this using viruses. We can do this either using a GFAP-emitting promoter that drives expression, usually, not all often, but not always, selectively in mula and then should shut off in photoreceptors. Or using a Cre-dependent flex AAV, where the insert is in backwards, Cre causes it to flip around and now express under a strong promoter. So these are injected intravitreally into the vitreous of the retina. And then what we're doing here, the data here is all done using wild type animals. So what we've done is to first label the cells with Sun1 GFP ahead of time. We then inject the virus, we apply NMDA, then we wait. And we're able to see that both INSM1, so the two of these viruses, one expressing the neurogenic transcription factor INSM1, the other expressing the neurogenic BHLHs E27 and NeuroG2, can induce GFP positive mulaglia to start to express OTX2. So this is a bipolar cell marker, so you can see here. This is pretty low level, but this is all happening in wild type animals. So what about inhibit? So second, we've been working to inhibit other transcription factors that repress neurogenic competence. And primarily, we have been focusing on factors in the Notch pathway. So HES1 as well as RPPJ and other components of the Notch pathway are strongly active in resting glia, as well as late-stage progenitors. So we globally disrupted Notch pathway signaling by knocking out RPPJ, which is the common transcriptional effector of Notch signaling. Again, selectively in mulaglia with a blast pre-ER, at the same time labeled these cells with the Sun1 GFP. So if we induce and then chase for 7 days, these are the mulaglia in control and RPPJ. They're essentially overlapping populations, but there are some big changes in gene expression. So first, we see that genes that are specific to resting mulaglia, such as glutamine synthetase, aquaporin-4, these are all downregulated mutants, as are Notch pathway Notch target genes, such as HES1, HES5, and ID1 through 3. Conversely, neurogenic BHLH factors, such as ASCL1, NeuroG2, and HES6, are upregulated. We can see more dramatic changes using ataxia. We see that the two populations at this age are essentially non-overlapping. We see that HES accessibility at Notch pathway, Notch target genes such as HES are down, whereas ASCL accessibility, ASCL1, which is basically inaccessible in the control, is now transposon accessible and able to be activated. So what happens to these cells? Do RPPJ deficient glia make neurons? They do, and they actually do in the absence of injury. So at 3 weeks in an uninjured retina, we don't see any neurons, but starting at 5 weeks and then rising through to about 4 months, we see a considerable number of glia. So we see that about 12% of the mulaglia give rise to OTX2 positive bipolars. About 3% give rise to HUCD positive amicrid cells. Following injury, we actually see substantially higher levels. And this just shows you some of the actual data. Now, can we improve this? So we can, and what I'm going to show you. And we can do this by viral misexpression of OCT4. So OCT4, as everyone knows, is a Yamanaka factor. Drives expressed in AES cells, drives pluripotency. When we misexpress it in control retina, basically in wild type retina, basically nothing much happens, nor when we misexpress it in the NFI deficient glia. But when we misexpress it in RPPJ deficient mulaglia, we see a massive increase in neurogenesis, about a 6-fold increase. This is about 5 weeks out. Cells basically are all bipolar cells, however, that are generated. And that you can see here, this is a single cell seq. from glia and glia-derived cells. You can see there's a whole lot more bipolars and fewer. These turn out to be resting glia. Now, what about if we combine NFI, the NFI mutants, and RPPJ mutants? Do we get better results? We get results that are actually about as good as it's possible to get, maybe a little too good. So here we have GLASC-3R, the NFI triple knockout, the RPPJ mutant, and then the SUN1. So we're knocking out 5 different alleles here, 4 genes and 1 reporter. So this is 3 weeks after NMDA treatment, 4 weeks after knockout. These are the glia and glia-derived cells. And you can see they're now moving all throughout the retina. Many are starting to express UCD, and even more are starting to express OTX2. And in fact, these 2 markers together represent about 80% of the GFP positive cells. And in only about 10% of the remaining cells are SOX9 positive glia. And you can see that this is actually starting to have some bad effects on the retina. If you kill the glia, the retina becomes dysmorphic. And that's actually exactly what we're seeing here. So this is about as good as, this is effectively, given the inefficiencies inherent in this process, this is probably close to 100% conversion of glia to neurons. Now, of course, this isn't useful as a therapy. This is a quadruple mutant. So what are some other ways that might be to target this that might be more therapeutically useful? The other thing I should say is that we don't see any proliferation in the RPPJ mutants. And that's fair enough, because Notch plays a critical role in controlling proliferation. What we've tried to do the third approach has been to target epigenetic modifiers and signaling pathways that repress neurogenic competence. So here, this is all work done using the NFI mutant mice. We've treated with small molecules that either are predicted to increase DNA chromatin accessibility, so 5-azacitidine, which is a DNA methylation inhibitor, or trichnostatin A, which is an HDAC inhibitor, or drugs that reduce inflammation, so STAT3 and NF-kappa B inhibitors. All of these roughly double the observed rates of neurogenesis. We're now working in combining these into polydrug cocktails. Now, what about proliferation? We've had very little success in inducing proliferation in glia, and it's no good to convert glia to neurons if you're depleting your glial population. Now, Muriel Perron's group here and Jerome with Jerome Roger and Ross Pochet's group at Baylor in Houston have shown that YAP signaling, disrupting YAP signaling in glia promotes proliferation. What we did here is that we used a dominant active form of YAP, which is inhibited by the Hippo pathway, and used this to infect selectively express in glia using a flexed AAV construct. We can see now when we label with EDU to visualize cells that are proliferating, we see a lot of EDU incorporation. We also see just a lot more glia overall. So this seems to be an effective way at inducing high levels of proliferation. So we've got essentially 100% neurogenic efficiency of making glia neurogenic, and now a way to make them proliferate. And actually, we did all this in the RPPJ mutant mice. So you don't need to have Notch in order to proliferate. And many of these cells actually express markers of OTX2 and other markers of neurons. So can we do better? We can still do better, but we've made a lot of progress. So we found multiple ways to promote proliferation and neurogenesis. Again, this is with viral misexpression. Some of these work in wild types. The OCT4, for instance, seems to work exclusively in the RPPJ knockout. We've identified RPPJ together with NFI factors as potent inhibitors of neurogenic competence and shown that different small molecules can also further promote this. Now, we're still mostly making bipolar and amacrine cells. So we have this third step to work on here, but I think so far so good. Now, in the last few minutes, I want to talk about some promises and pitfalls in applying these findings. And also, I want to look with a critical eye at some other claims to have developed methods to efficiently reprogram glia into neurons. Now, I think a lot of you are familiar with two papers that came out about 3 years ago now, one in Cell and one in Nature, that claimed that knockdown, so not even knockout, knockdown of a single RNA binding protein, a splicing factor called PTBP1 in glia, resulted in very efficient conversion of either muglia in the retina or astrocytes in the brain into functional neurons, and showed that this could recover deficits seen in a chemical model of Parkinson's disease. So there's a lot of excitement about this, but these claims were not very solid in the first place. First of all, the key claim here is that the glia turn into the neurons. You got to do that using cell lineage analysis. The way it's usually done in development is to label the cells before you do any kind of manipulation and to use a genetic label. These studies were all done using AAV-based mini promoters, specifically the GFAP mini promoter, to infer cell lineages. No transitional states were identified. You basically had perfect-looking glia converting into perfect-looking neurons. Nothing looked immature along the way. And then finally, there was no actual genetic loss of function here. This was all done using siRNA or antisense oligos. So as in a review I wrote with Josh, in a perspective piece I wrote with Josh Saines shortly after this came out, the optimistic interpretation here is that you're seeing cell conversion, but it may well be that what you're seeing instead is a misbehaving mini promoter. So that if you infect these cells, you see signal going down in glia and coming up in neurons. That may simply reflect the fact that the actual viral promoter that you're using isn't behaving well. Because again, these viral approaches infect neurons, glia, they infect all cells. And Chun-Li Zhang at UT Southwestern published a paper in Cell in 2021 that also cast doubt on this, that basically said that, yeah, GFAP-based AAV, when used to either overexpress NeuroD1 or knock down PTBP1, actually gave leaky expression, shut off in astrocytes, turned on in neurons. So we set out to investigate this more using genetic-based lineage mapping and loss of function. So in this case, what we did is that we both marked the glia, we marked retinal muglia using a SUN1 GFP and simultaneously knocked out PTBP1 and then waited to see if we got neurons. Now we can very efficiently knock out PTBP1 in the retina, but we don't see any neurons. We don't see any neurons either under uninjured conditions or following injury. So the previous claim was that basically you took out PTBP1, all the glia turn into ganglion cells, which in turn reconnect with the brain and are perfectly functional. We see nothing. Now, what about in fact, PTBP1 mute deficient glia remain glial. They don't downregulate any glial markers. What about in the brain? Well, so that work was published last year in Cell Reports. This is coming out essentially any day in Nature, where we disrupted PTBP1 in astrocytes using the ALDH1 CRER transgenic line. We see again efficient loss of PTBP1 in astrocytes, but no conversion of glia to neurons, no loss of glial markers. We've profiled these cells using single-cell RNA-seq, and the changes in gene expression are very minor. So this isn't going to do it. PTBP1 is not what you want. So what's causing these problems? Well, again, Chun-Li Zhang suggested it might be the mini promoters, and we wanted to investigate this more systematically. And we investigated this basically somewhat inadvertently by using GFAP to drive expression of a number of different transcription factors that we thought were strong candidates for promoting glial reprogramming. So here but what we did that was different was that we labeled the glia ahead of time using the glass crayon. So this is a prospective genetic lineage analysis prior to infection. So what we see is that the control vectors work fine, and so do few of the other factors that we tested, notably OCT4. But basically every other factor shut off in glia and then switched on in intraretinal cells, intraretinal neurons, bipolars and amacrines. So if we'd been doing this without a lineage analysis, we would have concluded that we were converting glia to neurons. And in fact, we were doing nothing of the sort. So this seems to be a problem intrinsic to the mini promoter. There's a lot of effort invested in using mini promoters to drive cell type specific expression. So what can we do to actually improve this? Because this is not going to be specific to GFAP. So why is this happening? It's probably due to the fact that both transacting factors, we're misexpressing transcription factors that may act on the GFAP mini promoter to change its specificity. Also, we're bringing in segments of DNA that may allow other transcription factors to bind. So yeah, mini promoters are not insulated from the broader environment of whatever happens to be in the vector. Now, however, there are ways to potentially make this better. So one thing that we have found is that, so this is work in collaboration with Ben Langmein's group in the Department of Computer Science at Hopkins, is that there's a very extensive range of cell type specific alternative splicing of genes that are very broadly expressed. So this shows you a couple of examples of splicing of exons that are specific to neurons. So this is mouse specific, this is in PLS3, and in human specific exons as well. So these tend to be broadly cell type specific and well conserved evolutionarily. Now, we asked whether it might be possible to use some of these alternative splices, alternative cell type specific exons to drive in the context of translational frame shifting, to selectively drive expression of different reporter constructs. So what we have here is essentially, we can use any promoter to do this. We then have a cell type specific exon that then frame shifts to express EGFP, whereas the more conventional ubiquitous splicing pattern gives rise to DS-RET. Now, splicing is relatively insensitive to whatever's outside your exon, your alternative exon sets. This might work much better in an AAV-BIS context and can potentially be combined with mini promoters in a Boolean fashion to give highly cell type specific expression patterns. So these are three examples of the so-called splicing linked expression design, or SLED vectors. This is one where we've taken a ubiquitous promoter and used it to drive pan-neuronal expression using this PLS3 intron, as shown here. Basically, we see very high expression just in neurons and not in glia. We can do the same for photoreceptors. So this is a photoreceptor specific exon cassette with a ubiquitous promoter infected into the retina, very specific expression of photoreceptors. And this is an example of how we can make this Boolean. Here we have an alternative exon that is specific to excitatory neurons in glia. So we can combine this with a pan-neuronal H-syn promoter and end up generating an excitatory neuron-specific construct. We can use these to misexpress essentially any genes that we like. the main challenge here is simply keeping this splicing construct relatively small. So I'd like to close now. Thank the folks who did the work, or thank the folks in the lab who've always make it a fun place to be. Thank our sources of support. And specifically, I would like to thank Tan Huang, who now has his own lab at the University of Michigan, and Jay Wang, who now has his own lab in Guangzhou, who began who carried out the published studies that I showed you first. In particular, Niet Le and Haley Apple have two current grad students in the lab, have done much of the unpublished work, and John Ling pioneered the sled-based studies. I'd like to thank our collaborators, and we put all our manuscripts on bioRxiv prior to publication, and I announce that on Twitter. So if you want to keep track of what we're doing, please follow us. Thank you very much.